Good afternoon, ladies and gentlemen. Welcome to the Oxford Cannabinoid Technology Holdings PLC Investor Update and Q&A session. Throughout this recorded presentation, investors will be in listen-only mode. Questions are encouraged and can be submitted at any time using the Q&A tab situated on the right-hand corner of your screen. Just please simply type in your questions at any time and press send. The company may not be in a position to answer every question it receives during the meeting itself. However, the company will review all questions submitted today and publish responses where it's appropriate to do so. Before we begin, we would like to submit the following poll, and if you'd be so kind as to give that your kind attention, I'm sure the company would be most grateful. And I'd now like to hand over to Clarissa Shoei-Mimo-Coker, COO. Good afternoon.
Thanks, Mark, and hi, everybody. Welcome to this Investor Meet Company Q&A session with Oxford Cannabinoid Technologies. I'm Clarissa Shoei-Mimo-Coker, as Mark just said. I'm COO here at OCT, and it's a pleasure to be here today with our CEO, Dr. John Lucas, and our CSO, Dr. Valentino Paravicini, to talk to you about OCT and answer your questions about the company. Just to give you a bit of a heads up on the format of the session, firstly, I'm going to give a brief intro to the company for those of you who might be maybe new to our story or perhaps need a little refresher of who we are and what we do. Then I'll be handing over to Valentino, who's going to take us through a deeper dive on our 4 drug development programs, which is an answer to some questions that we've received from shareholders in advance of today's session. Then we'll hear from John on what the future of OCT looks like. And finally, between us, we'll address any new questions that are submitted during the session, as Mark just explained. So please do make sure you submit your questions along the way, and we'll get to as many of them as we can in the time we have together. Now on to my brief introduction to OCT. Well, we're a pharmaceutical company who listed on the main market of the London Stock Exchange on the 21st of May, 2021, so just a little over a year ago. We're developing next-generation non-addictive pain medication to replace opioids. From 2006 to 2020, the number of opioid prescriptions in the United States dropped from 215 million to 142 million. This trend tells us that doctors don't want to prescribe opioids, but we still don't have a viable replacement, and as a result, we have a lot of patients in pain. This is the patient group that we're targeting. These are patients with chronic and severe pain indications, representing an underserved market worth over £40 billion. We're not doing medical cannabis, which can't be approved by the FDA or the EMA as a licensed medicine, and which can't make any formal claims to treat any disease or conditions, and which isn't reimbursable by insurance companies and can't generate market exclusivity. We are different. We're a pharmaceutical company in name and nature, and we're taking our drug candidates through robust clinical trials for specific pain indications, which are designed to give OCT regulatory approval, allowing doctors to prescribe and insurance companies to reimburse. Our model also allows us to improve the structure of cannabinoids, creating medicines with market exclusivity. Our aim, ultimately, is to provide new solutions for underserved patients suffering from severe and chronic pain. That's my brief overview done. Now over to Valentino for a deeper dive on our 4 drug development programs and the answers to your previously submitted questions. Over to you, Val.
Thank you very much, Clarissa. And thank you very much for joining online. As you know, since from the beginning, we decided to enlarge and expand our pipeline, and doing so to basically provide multiple shots on goal for the company. Our drug candidates target different receptors with different mechanism of action, thereby giving us the opportunity to target different diseases and different indication, and of course, different patients. That is the most important thing. I will go through our programs one by one in detail. Starting from program number 1. This is what we call our lead compound, OCT461201, which I will refer shortly as 201. This is a CB2 agonist, meaning it activates the CB2 receptor. It is the spearhead of our cannabinoid research engine. This is a licensed compound, and the preclinical development is fully underway. We are initially targeting neuropathic pain and visceral pain, and as I will show, this is aimed to begin a phase 1 clinical trial in Q1 2023. And it comes with NCE exclusivity in terms of IP. So to go more in the detail of what we're doing, we are a virtual company, and our way of delivering science and value is to build a strategic networking across different companies, a different background. Particularly after the IPO, we expanded our network of collaboration with both top-tier CROs, but also bespoke and boutique companies that are allowing us to deliver large packages or specific goals for the specific programs. And you can see some of this here on this slide. So one of the questions is why CB2 agonist? What is the rationale for drug development for a highly selective CB2 agonist or activator? Well, there is a large set of preclinical data from scientific literature. We've generated our own data sets, but also there is an availability of some clinical data. In the yin-yang of the endocannabinoid system, where you have CB1 versus CB2, we are convinced that targeting CB2 will provide therapeutic potential without causing psychotropic effects, and this is very important. Focusing on a single target receptor has the advantage of reducing the risk of drug-to-drug interaction, or DDI, as you may have heard, and toxicity. By selecting CB2 over CB1, the drug candidate is aimed at patients who want to avoid psychotropic effect or they cannot afford to have it. CB2 activation has an anti-inflammatory component as well, and this may be important for immune disorder, but also as a double-pronged approach for tumor. On one side, it will reduce tumor growth inhibition directly. On the other, it will allow the immune response to attack the cancer cell better, and therefore to improve the outcome. CB2 gives protection in models of neuropathy as well. And most importantly, as you can see, it provides a beneficial effect in analgesia and visceral hypersensitivity, as I will show you in a moment. So OCT461201 was licensed from Ascot Incorporated. It's a company in Japan that its origin in Pfizer. 201 is a highly potent selective agonist or activator CB2, as I mentioned, and the compound was initially developed for irritable bowel syndrome, particularly the one characterized by diarrhea, so IBS-D. We have since expanded the indication to include the chemotherapy-induced peripheral neuropathy, or CIPN. We start, as I mentioned, a phase 1 trial in Q1 2023. The exclusivity here comes from regulatory data exclusivity and patent protection that come with the compound. And we're also aiming to generate new as we go through the preclinical package. Basically, at a glance, there's no THC-like effect, no psychotropic effect, but we do have a targeting of the CB2 receptor in the central nervous system. And this is an important differentiator from other compounds out there. For example, lorinab does not have good CNS exposure. There was a compound that was trying to develop by Arena Pharmaceutical for IBS. The phase 2b trial wasn't successful. We do know not exactly why the trial failed, but certainly the lack of CNS exposure might be one of the reasons. In our case, we do observe exposure. We do observe efficacy in animal models at low milligram doses in IBS, and as I'm going to mention, in other diseases, and crucially, no adverse effect at a high dose. The compound has drug-like properties, excellent PK profile in rat, in dog, and excellent pharmacotoxicity, both in vitro and in vivo. To support the indication for the treatment of IBS, the FDA recommends primary endpoints relevant for at least 2 IBS signs and symptoms. One is abnormal defecation, and the other one is abdominal pain. At the time of the license, the existing ASCAT data package show both of them. As you can see here on the top left, we see visceral pain reduction, and also on the right, top right, the reduction in water content. This is dose dependent, as you can see, so it goes up with the benefit goes up with increasing the dose. And quite interestingly, I would like to say, keep in mind number 3 and 10, 3 and 10 milligram per kg. I will come back to later to that in my presentation. And particularly, as you can see by the asterisk, this is statistical significant difference. But as you know, it takes 2 to tango. Efficacy and safety are both paramounts in drug discovery. And at the bottom right here, you see that at 300 mg per kg, 201 is safe and tolerable. There's no drop in body temperature, but also, as shown in this graph, but also there's no stupor, there's no dizziness, no other effect really. And keep in mind that at 300 mg per kg, the amount of compound in the blood is more than 480 times higher than you do see at efficacy of 3 mg per kg. So basically, the dose escalation is possible with no adverse effect. Importantly, and I want to stress this, all the in vivo data that are showing here and the one I'm going to show you later are obtained using the compound via oral route, which is the chosen route of administration for patients. So really indicative of really what happened when we go into the clinic. Now, this slide is a bit complex. There's a lot of writing, but you need to focus basically on the main points here at the one right. And in summary, as you can see, 201 is a highly selective agonist activator. You see the table here, 4,000 over CB1 receptor in human. This is important, as I mentioned, because there's no psychotropic effect for patients who do not want or cannot accept that. The multi-pharmacology, we were asked about that, can be useful, but we will exploit that in other programs. Here, we want to be selective because it's important not to have multi-pharmacology that may have detrimental effect when it comes to adverse effect. Focusing on a single target receptor has the advantage of reducing the risk of drug-to-drug interaction and toxicity. Reducing drug-to-drug interaction is important because it means that once approved, OCT461201 offers greater potential for use in combination with other drug, either for the same indication or just in case a patient is taking out a drug for other indication. Sorry, going back, I want to say that the compound has excellent solubility. Low solubility can be a massive problem when you do drug discovery and development. There's no cardiotoxicity, no genotoxicity liabilities, and there was a positive safety and cellular pharmacology. We tested 64 different receptors to the risk liability, and all of them were clean. So these were the data that we had already at the time when we licensed the compound from Ascot. But we wanted to do more. It was important that we knew whether 201 had a good safety profile in pharmacokinetic species. I show you some data with rat, and now it's important to show that in dog. So we actioned an exploratory toxicity study. The animal received the drug at quite high doses, 10, 20, and 30 mg per kg, and for 3 days. And here we show that the compound can be found at high concentration in the blood, consistently across all 3 days. I'm showing only day 1 and 3, and it is over the projected efficacious concentration. So basically, we are always over the concentration that create benefit potentially for the patient. There is good Cmax. The area under the curve, that basically this area under the line, is very good, meaning that the compound stays long enough in the blood, potentially allowing us to give only 2 doses per day or BID rather than more. Importantly, I have to add here, there's no adverse effect observed, and even in 4 histopathological analysis of many tissue in the animal, we saw no effect, no dangerous effect. But we knew that 201 was good in IBS, but strategically, obviously, we wanted to do indication expansion, meaning we want to target more disease, meaning we want to help more patients, meaning we have more shots on goal. The best thing that you can do in R&D is following the science and adapt and evolve based on the science. And that's what we did at OCT with chemotherapy-induced peripheral neuropathy, or CIPN. CIPN is a disorder that is observed in a high percentage of patients undergoing cancer treatment. The drug damages the nerves, particularly the small fiber nerves. And even a gentle touch or the change in temperature can cause intolerable pain. Sometimes it is so bad that the patient stops the treatment. That is the only hope that they may have. So what we did, we used a model for the CIPN. We treated the animal with paclitaxel, which is a common oncology drug that is used in human, for example, for triple-negative breast cancer. And this drug causes CIPN exactly in the rat, exactly like in human. 201, as you can see on the top, reduces significantly the pain caused by touch, and also with very significant difference, as you can see by the statistical analysis, but also at the bottom, particularly with the pain caused by cold. The benchmark of every new drug candidate is the standard of care. In case of CIPN, there's no drug product approved for CIPN, but our compound is certainly basically showing equivalent activity to one of the off-label drugs that are commonly used in therapy, in this case, pregabalin. And as you can see, I asked you earlier to remember 3 and 10 mg per kg. The number returns here at 10 mg per kg. The compound is basically identical to 30 mg per kg of the drug that is normally used for CIPN. So very good data. Interestingly, the damage of the small fibers that you see in CIPN can be seen in other disorder. For example, in painful diabetic neuropathy, which is observed at the early stage of diabetic polyneuropathy. So this provides another potential target indication, which we will explore in due course. So in order to accelerate our development for 201, we decided to enter an integrated clinical development platform called INDIGO, or INDIGO, referring to IND process in the United States, FDA process, at Evocate. Basically, this is a package that is designed to do all the steps that we allow a company to get to clinical trials, going through the process of chemistry development, familiarization, scale up, non-GMP and GMP manufacturing, so good manufacturing practices, but also formulation development and stability to get the drug ready for the clinical trial in a faster way. This can be customized based on the need of OCT, and that's what happened. And what is important is that all the activities, meaning drug substance, drug product, the preclinical safety, and the pharmacotoxicology, but also the regulatory path, they go in parallel. And the result, they are submitted in an electronic tablet called SEND. That means it's ready to regulatory submission to the FDA. Now, I ask you to focus on this sort of central panel or part of the graph with all this Gantt chart with all these bars, basically. And now I'll move to the next slide, because those bars are represented here, which this line, and each of them is a different experiment that we have performed, the one in green, or the one that are going to be performed, the one in black. Actually, today we got a result for in vitro plasma protein binding in 3 species, and it is actually quite good as well. So all these data show that the compound is, in terms of ADME, general toxicology, general safety studies, and general toxicity studies, is going to be safe and good to use in the patient. And all the green, as I said, are good. I'm going to mention a bit about, touch on upon the general toxicology studies in animal soon. So moving to another to important element of INDIGO platform, on the top you have a snapshot of the chemistry, manufacturing, and controls, or CMC activities around the drug substance on the top and the drug product on the bottom. So we're looking at manufacturing, we're looking at analysis of the crystal, we're looking at a formulation. Because an active compound, it is not a drug product per se. The question you have to ask yourself is, can you manufacture in a large scale? Is it stable in the form that you manufacture it? Can we make it so that it goes into solution? Can we formulate it so that the patient can actually use it in a good way? So our work will answer those questions. Some of them have already been answered, and they're good. And this will allow us to turn the drug substance, 201, into actually a drug product, package and label and ready to use in a clinical trial, initially, obviously, for the healthy volunteer. But this obviously can happen only after regulatory approval, and then can be also streamlined through the INDIGO process, as you can see at the bottom here. However, it is likely that we transfer these packages to the clinical research organization who support us for the clinical trial. And speaking of the clinical trial, we're now ready to move into a clinical phase and start the clinical phase. I told you the 201 has a very selective pharmacological profile. Yes, it does. Have an excellent in vitro pharmacotoxicology profile. So far, yes. Efficacy in vivo, yes, as well. In 2 models of pain, significant analgesic effect at both 3 mg per kg and 10 mg per kg. And no adverse effect observed with multiple doses in dogs. I show data in 3 days, but now I can tell you that 7 days at 100 mg per kg in dog and 300 mg per kg in rat show no toxicity whatsoever. And also this is through an initial toxicology study. It means that difference histopathology. It means the difference between the active dose and the toxic dose is very high. And this is very important when you're developing a drug. Obviously, in the clinical development, we will aim to do a single ascending dose first with adaptive design, which may include an integrated multiple ascending dose into it. We now ask 7 companies to provide the proposal. 5 were formally investigated. 3 was selected for a face-to-face bid defense meeting. We already have this meeting. We selected the winning bid, and now we are making sure that everything is in shape so that we can approve the statement of work and obviously signing the contract agreement. And the current timelines are aiming for a first patient, first dose in Q1 2023, following the clinical path that we developed together with VCLS, as you see at the bottom right. We move now to program number 2, OCT461201 or 401 for short, is our second development program, and is based on a combination of phytocannabinoids. We are initially targeting trigeminal neuralgia, and the aim is to begin phase 1 in Q4 2022, so at the end of the year. Exclusivity will come from orphan drug designation in this case, but there are also technological elements, and I want to stress this because I'm going to talk about some of them, based on the particular formulation and then the drug device combo that we're using it, which may add additional unicity to the product. Once again, at the center of our effort is a network of companies that can provide specific activities and overall complete packages. OZUK and H2 Prespar are leading the way into the respiratory device development. CRL, Charles River, for preclinical development. And then you have other companies that are supporting the regulatory and clinical activity as well. And basically what we're doing, we are harnessing the yin-yang of phytocannabinoids, THC and CBD, to develop an API combination and a medical device, and effectively a drug device combination that, as I say, is targeting trigeminal neuralgia. (401), as I say, is a PCB phytocannabinoid drug candidate. Also, orphan drug designation will provide exclusivity and also expedite route to market. And this is crucial as well. Additional exclusivity may arise from the formulation and the technology we are developing. We are performing a common efficacy study. There's the aim to confirm the wide breadth of data that are available for preclinically and clinically as well. Trigeminal neuralgia provides also with an homogeneous population of patients, which will allow OCT to power a better clinical trial and optimize for success for OCT and, more importantly, for the patient. And the inhalation route will ensure a faster onset of the benefit, a lower dosage, and potentially less adverse effect. Here on the right, HT Prespar were really nice to show us a prototype of when our device may look with the OCT colors in it. So I keep saying this, doing the right thing for the patients means a lot. It's central to our activity. And in this case, means delivering a metered and consistent dose. Our pressure metered dose inhaler, or PMDI, that's similar to the one I use for asthma, for example, is a medical device, proper, enabling better absorption, improved bioavailability, and, as I mentioned, faster onset. OCT partnered with first-tier expert in PMDI development to achieve optimal formulation, accurate dosage, and to have a consistent biodelivery across the lifespan of the device, from day one to the end of the formulation. OZUK, HT Prespar, and Aptar, from the technological aspect, are supporting us. Charles Rivers for the in vivo preclinical package. VCLS is our regulatory consultant to provide regulatory strategy and a way to clinical path. And again, other company, for example, Purisys, is providing the API that comes with the drug master file, meaning it's already useful to go into supporting our application to the regulatory agencies. So technological performance is the key component. I mentioned that, so the success of 401 program. We partner with OZUK to make sure that the dosage was constant, that the particle size, meaning the size of the particle in the puff that you generate when you actually press and actuate your device, is allowing the entry into the lung, but not the exit when you expire. And it was also important that the compounds, the API, are stable, stably maintained, even at the really vaccine condition. And we did a lot of work in formulation that is really summarized in this slide, but there's a lot of work behind it, to identify crucial but not obvious excipients, which enable both long-term stability, as you can see here in the red box. So you can see basically after 3 months at 40°, so well over room temperature, you can still have 100% of both CBD and THC. That is better than the starting formulation that we had at the beginning. So that's quite important. And what it means is that the patient can use the drug device combo with confidence wherever they are, depending on independently from the climate, getting 100% of THC and CBD the last day as well as the first day of their usage. All this work has allowed us to generate a tox batch that will be used actually in the toxicology study at Charles River, which has the same characteristic of the clinical batch. This characteristic I highlighted here on the right, what we call the specification of the drug device. They're showing all the parameters suggested by standard operating procedure or SOPs, and also by the Pharmacopoeia Europea or PHEOR. We already chosen the canisters you see in the bottom, the type of canister, the type of valve, the device, and the concentration of the active compounds and the formulation as well, as I mentioned. The combination allows us, as you can see on the bottom left, to obtain a stable delivery of the compounds, or the active compounds, at the right dose, at the right ratio, from the first actuation. That means actually the path that you do when you press the device, from 3 to 150. So that's well over the required 120 that is suggested, or actually required in this case, by the Pharmacopoeia Europea. So we are really in good space for that. By way of a further update on the status, as I mentioned, the preclinical and clinical path have been drawn with support by VCLS, as you see on the top right. In terms of the preclinical package, we're proceeding with the aerosol analytical method and the plasma analytical method as well at Charles River. MD Bioscience will perform the efficacy and PK study. And the full preclinical vivo pharmacotoxicological package, we start in June in rat and July in dog. And then this again will be reported in SEND format for the regulator in the US and EU. This format, by the way, is not required in Australia, where we will perform our clinical trial, because the HREC path will be supported by a latter package enabled by the 7-day toxicology only, plus the body of preclinical and clinical evidence that is publicly available when it comes to THC and CBD. We've chosen the GLP batch manufacturer for the UK and the preclinical studies, as I mentioned. And the GMP manufacturer, again, we are waiting in the case for contractual agreement. On the clinical package side, obviously, we soon sign a CRO to support our activity in Australia. First with the protocol design, then the patient recruitment and the regulatory ethical approval, followed by the phase 1 as such. The trial is designed to have 2 parts, a single dose ascending dose, SAD, followed by a multiple ascending dose, MAD. Our aim is to complete the SAD in Q1 2023 and the MAD in Q2 2023 as well. Now, in the next slide is just a visual representation of what I just talked to you through the slide 23. This is an overview. We have the Gantt chart here focusing on the drug device activities on the top, on the clinical path through the HREC in Australia in the middle. And then at the bottom, we have the preclinical toxicological studies that again, they will support the HREC, although the HREC per se will not require a full package to start the SAD. And we move now to next programs really, 3 and 4. They're both stemming from the same starting point. That is the collaboratory network created by OCT to expand its existing library and provide a seamless development from design of the compound, chemical design, the manufacturing, and the translational assay, both in vitro and in vivo, for multiple drug candidates. Program 3 and 4 are targeting the endocannabinoid system in different ways. Program 3 will target both CB1 and CB2 at the same time, so multiple pharmacology. Program 4 is targeting an orphan ECR receptor called GPR55. But the starting point for both, as I said, is the Canopy library, as I'm going to show you in the second. In fact, more than that is our combined library, if you want. At IPO, some of you may remember, we had a library of about 100 phytocannabinoid derivatives. They were proprietary to OCT. And we wanted to expand that to 200 what we call next generation cannabinoids. I'm happy to report that we have done more than that. We combined the OCT library we already have with the Canopy Growth in License library. The compound came with an IP covering the library with 14 patent family and a large data sets allowing us to move from lead and then enter into candidate selection space faster. We're aiming to do that within the year. The collaboration with Ariada, which developed the Canopy library in the first place, has allowed us to expand even further the library. And we have now additional 36 new OCT proprietary compounds. So these are an addition of the in-license from Canopy. So we now have 450 compounds, which is basically more than double our commitment initially. And this is really showing the importance of the synergism in OCT cannabinoid research engine. These compounds add better bioavailability. We can use prodrug approach. We expanded our pharmacology to CB1, CB2, GPR55, and as I say, multi-pharmacology. So we're harnessing the activating multi-pharmacology that someone mentioned and asked for that. But also we are blocking GPR55. So it's an antagonist in this case, we call it. This is allowing us to target alternative pain indication, but also neurological condition, such as epilepsy, and potentially other indication in the oncology space later, perhaps with strategic alliances with other pharmas. What do we do? How do we approach with programs? This is complicated, but basically, one thing important is that the phytocannabinoid receptors and the derivatives are targeting a quite complex biology of the endocannabinoid system, particularly the so-called G protein coupled receptor, GPCR. CB1, CB2, GPR55 are all GPCR. This receptor has the ability to change the cell activity using different pathways, as you can see at the top right here. So if you evaluate only one of them, you really risk to throw away the baby with the dirty water, or to have a false positive, meaning something that actually doesn't work, but it seems like. At Dariata, OCT can evaluate the capability of our phytocannabinoids to target different biological signaling, as you see in the table here on the top. And the smaller the number, the better it is actually, the lower the dosage that you have to use. Then we check the pharmacokinetics when we establish that the company is working in vitro in different pathways. Then we check the pharmacokinetics and brain penetration data in vivo already. This allows us to basically tells us if the compound behaves like a potential drug in vivo. The next step that we move to inter vivo, evaluate the in vivo analgesic effect using the tetrad assay that as you can see here on the bottom right. And inter vivo can also perform other relevant models for other indication. I mentioned, for example, epilepsy, and in vivo is also all the three clinical relevant epilepsy model in rat. So zoom in a bit more on program 4, and the previous slide applied for both program 3 and program 4, by the way. Our quest for the next generation cannabinoid drug, I think, is going in the right direction. We identified OCT proprietary compounds antagonizing GPR55 signaling, activated using by 3 different activators. This is basically validating the basic assay that we use in the beta-arrestin assay. So not only one type of activation, but 3. And it's providing, obviously, additional confidence that our company is working. Further validation has been provided by checking another of those signaling pathways that I mentioned before, the phosphorylation of a protein called ERK, which is importantly, and it's performed with a homogeneous assay, so quite relevant and reliable. And again, Tanso, is a company in Japan, will provide an orthogonal assay to validate even further our results. But what about the possibility, actually, that our derivatives can inhibit neural cell? Can they inhibit the firing of the neuron cells there, in order to reduce the pain and potentially reduce epilepsy? As you know, the sensation of pain and epilepsy is caused by unnatural or excessive firing of the neuron cells. So we use a translational model that has been validated in clinic by the likes of Pfizer, for example, and this is actually innovation in innovation. We use induced pluripotent stem cells that are differentiated, or we say neuralized, by our collaborator StemTech. And then they're plated in this special plates. They have these blue lines and blue dots are actually electrode. We also add astrocyte. That is to mimic even better the CNS. So this is a multicellular assay, it's not just neuron. Astrocytes are present around the neurons in the central nervous system. The cells start to form synapses. I don't know if you can see the red dots on the top right. And at this moment, they start firing spontaneously. And the electrode, they measure this activity, this firing, with these spikes. It's really actually almost like an electricity signal. And the question is, can we block this? And if you use the right drug, yes, you can. And in case of OCT, we do. So here at the bottom left, we have analogs, the CBG analog from our library and the CBD analog as well. The basal line is the dotted line. That means normal activity, this spike in here. Or every dot underneath means that we are reducing the activity. And as you can see on the right, what we're looking among these many dots and many compounds is one like D3CAN1 that will allow you to have inhibition of the signal in a dose response way, similar to the SOCs. And this is exactly what we are observing with several of our analog. In fact, StemTech was just doing this basically as bread and butter. They say they were really positively impressed by the number of hits from our focus library. So I really say that we achieved excellent results and very quickly, even in program 4. So at this point, I think my presentation is over. I gave you an overview of all the 4 programs, and I hope that I answered most of the questions that were asked before this meeting. And I'm going to pass the baton to John in the next slide.
Thank you, Valentino. So now that you know more about our drug development programs, I'd like to return to the investment rationale. OCT is not a medical cannabis company. We're a pharmaceutical company who happens to focus on cannabinoids. And we're focusing on cannabinoids and their receptors because we believe cannabinoids offer the best opportunity to reduce or replace opioids in many indications. Our pipeline is diversified to balance risk and offers multiple opportunities for success. And although we develop phytocannabinoids, that's in our program 2, our research, as you can see, programs 3 and 4, and our lead candidate, we're focused on the next generation of compounds with superior properties like selective targeting. And OCT, we are a small company, but because we've built a network of research and clinical development partners, we now have the same capabilities as a big pharma. One of the questions asked was, around our cash position, and this is very important for us as well as our investors. Our cash position, our current cash position, takes us to the end of March 2023 if we don't cut any programs or delay anything or take any further actions. We're going to need to raise before then to continue our programs, and the amount we raise is going to be dependent upon market conditions. We may or may not be able to take all of our programs forward beyond what we stated in the prospectus. We expect the market conditions to improve over the summer and our share price to reflect the transition from a preclinical to clinical stage pharmaceutical company. However, we may have to be selective in the programs we advance until market conditions improves. Another question that was asked was around, why I haven't purchased shares. Well, that's not for the lack of trying. I guess, the answer is I'm doing my job because I'm always looking for opportunities, and that, unfortunately means I'm usually in possession of potentially price sensitive information on a continual basis. And, I guess that, as you can see in the deal we closed with Canopy and the other advances that we've made, this is paying off. However, unfortunately for me, it keeps me in closed period. But this concludes, so at this point, this concludes our presentation, and we're going to be happy to take any questions. I'll turn it over to Clarissa to moderate.
That's great. Well, let me just jump in the meantime, just to give you a few moments to review the questions submitted already. But thank you, John, Val, and Clarissa for your update this afternoon. Ladies and gentlemen, please do continue to submit your questions using the Q&A tab situated on the right-hand corner of your screen. But just while the company take a few moments to review those questions submitted already, I'd like to remind you that a recording of this presentation, along with a copy of the slides and the published Q&A, can be accessed via your Investor Meet Company dashboard. Clarissa, if I may, obviously you had a number of questions that were pre-submitted. I know you've taken the time to address a number of those throughout today's presentation. You've also had a number that have come in through today's live meeting. But perhaps if I may just hand back to you and read out those questions and give a response where it's appropriate to do so, and I'll pick up from you at the end.
Thanks, Mark. Yeah, absolutely. Okay, thanks, John and Val. We have got loads of questions in the Q&A, so we'll get through as many as we can in the time we've got available. To start with, I've got a question for Val, actually, which I think is going to be a very quick one to answer, because even I can answer this. It's even a science question. Val, one of the questions in the chat is, who is developing the pressurized metered dose inhaler for OCT430401?
Yes, so there are several companies involved. Mainly is a company, Oz UK. The people at Oz UK, they're stemming out from Vectura, actually. There were people that were there from the founding, and they moved really literally just across the road. They have 20 and odd year of experience in developing this inhaler. In fact, I cannot say, but a lot of the inhaler that people are more familiar with, they were developed by the scientists there. They're really good. They're very thorough in what they do. And they're really on the top, and they have a particularly good collaboration, and this is important for us because we basically generate really synergy with other company like HT Presspart that will actually provide the material, for example, the canister where we actually put the compound, and Aptar, that is the company that is providing the valve. The valve is the one that actually allow the compound to come out, if you want, from the canister, be given to the patient. HT Presspart will also giving us access to their device, that is the blue sort of plastic device that I show you. It is the one actually allowing also a proper dosing through the phases that are used by the patient.
Thank you. That's helpful. John, I think a question for you next. One of the questions that's come up from a few different people, actually, and it's phrased in slightly different ways. So I'm going to take a liberty and sort of and phrase it to encompass everybody's question, hopefully. People are saying, hey, look, this is pharma and it's high risk. So isn't there quite a big risk attached to the approach that OCT is taking? Maybe you could just talk a bit about that.
Sure. Well, there's a significant risk to pharmaceutical drug development that's inherent. However, where there's risk, there's opportunity. And our strategy is to lower the risk and seize the opportunity. And we believe we can lower the risk by developing cannabinoids. That's the whole point of the business, because I think everyone knows that cannabinoids have been used for thousands of years successfully to reduce pain. And we are also lowering our risk through over 75 years of experience, combined experience and executive team. We know how to develop drug products. And finally, we don't fight the science, we follow the science. If the data says something is not working, we either fix it or move on.
Brilliant, thank you very much. Val, I've got a question for you, and I know you're going to like this one because I've asked it to you before. It's one that's just come in on the Q&A. I hope you're going to answer it in the way that you have to me before, because it's a lovely answer. So which of all the options that we have in front of us excites you the most? Which one are you most excited by? I'm pretty sure I know what you're going to say.
The answer to say is like asking which is your favorite child. These all of these are really good programs, and they are exciting in different ways. One is the one that we receive already sort of grown up a bit, if you want, and we're growing it like a teenager. The other one will really accelerate teenagehood and really going to adulthood with this program too. And there is the buzzing really there in terms of the potential THC and CBD and the inhaler. This is really going to be important for us, because the benefit it can provide being inhaled is great. And program 3 and 4 are really novel. I mean, we have these compounds, with a set of compounds. We enlarged them with a canopy deal that was excitingly said. We have this amount of data showing that this compound can do well. We just had to improve it slightly bit, because not every compound that works in vitro can work and then in vivo, and that's what we are doing. And we are already progressing. We already see data in vivo. And we can choose with pharmacology. I mean, we decided to go for CB1, CB2 double agonist because it was important to sort of harness both, as I mentioned, the yin yang of ECS, but also the new program 4. I mean, it's great. I think it can be really revolutionary, both for epilepsy and in potential oncology. So I'm really excited about all of this.
So you can't choose one. That's fine. You can't choose one. No. Thank you, Val. John, one for you, I think, this time. People talking about the next fundraise and asking about, who we might be targeting, hoping that institutional investors will be on that list. Is that something you could just speak to?
Yes, we do aim to attract institutional investors, OCD. We think this is very important. The current shareholder base comprises predominantly retail, high net worth family offices. But as we grow and need to raise further funds for clinical development and facilitate commercialization of our first prescription medicines, we're going to be seeking institutional financial backing.
Makes sense. Thank you. I think probably just one last question. So I've got my eye on the time. We're due to wrap up shortly. People asking about, you did speak on the investment rationale just at the end there, but perhaps you could just talk again, John, about what makes OCT a compelling investment opportunity kind of right now, particularly looking at the struggles with the share price and the biotech sector has had over the last 6 to 9 months.
Well, we're a milestone-driven company, and I think, what you have to look at is the milestones that we said that we were going to reach, and that is what we're going to do. We are based on milestones, and we have a diversified pipeline, which reduces risk. We've created this network of all the partners. We did a lot of heavy lifting over the last year that gives us the same capabilities as a big pharma, so we can do anything that a big pharma can do. But however, unlike a large pharma, we are working at unparalleled speed to develop our drug candidates. For example, look at the timelines. For example, we have for program 2, we announced preclinical development was starting, I think, like in October, and we're going to be in the clinic a year later. And it's a similar timeline for OCT461201. So, these are unheard of speed at which we're working at. And I think, I would encourage investors to look at how long that it normally takes to take a program from discovery and into the clinic. And one of the advantages that we have with working with cannabinoids is that, is that background on the profile that allows us, that's been, showing to be true, that we can develop these very quickly.
That's great.
Yeah.
I'll jump in at this point then, Clarissa, and thank you very much for hosting the Q&A, and to John and to Val for your responses. And thank you once again to all the investors that have taken time to submit questions. And of course, we haven't gone through all the questions, but we'll make sure that the company has access to these post today's meeting. And we'll publish those responses on the Investor Meet Company website. John, I was going to ask you for some closing comments, but I think you've just done it perfectly. So for fear of asking you to wrap up again, I'd like to thank you all once again for your time and for your presentation this afternoon. I know investor feedback is important, so I'm going to now redirect investors to give you their thoughts and expectations. So, ladies and gentlemen, if I may ask you not to close this session, as I'll now redirect you for order so that you can provide your thoughts and views directly to the company. On behalf of the management team of Oxford Cannabinoid Technologies, I'd like to thank you for attending today's presentation, and that concludes today's session. So I may thank you all once again for your time.
Thank you very much.
Thank you.
Thank you, guys. Thanks.